Today we will be exploring the way that the central nervous system interacts with and controls target organs. And so we're going to be exploring the neurons that go directly from the spinal cord or the brain and eventually approach target organs in the autonomic nervous system. And it's helpful to kind of set it up so that we're looking at both branches at once, the sympathetic and the parasympathetic branches of the autonomic nervous system. To be clear, the ANS, the autonomic nervous system, is a branch of the motor branch of the peripheral nervous system. And it being a motor branch, that means it's a descending branch. We're bringing information from the brain, from the spinal cord in, and allowing us to then control these different target structures. But importantly, it is autonomic, which means automatic, which means involuntary. We are going to write sympathetic on the left side of your paper. The sympathetic branch of the ANS, the autonomic nervous system, is associated with things like adrenaline or epinephrine and noradrenaline or norepinephrine. And this is famously known as the fight, flight, or freeze branch of the autonomic nervous system. And the four kind of physiological events or categories of events where the sympathetic nervous system would take over would be things like emergency, exercise, excitement, and embarrassment. So 3 F's, 4 E's. On the right-hand side, we're going to write parasympathetic. The parasympathetic nervous system is less well known. The primary neurotransmitter here would be acetylcholine or acetylcholine, although it works differently from the way it works that we've discussed in the past, where it's this excitatory muscle contraction kind of a neurotransmitter. It's going to be a little different this time. The parasympathetic nervous system is associated with digestion, diuresis, which is urination, and defecation. So this is going to be your digestive system kind of a, there's other things that it does, but that's a primary thing that the parasympathetic nervous system is in charge of. And then we think about the parasympathetic nervous system being most active during physiological events such as, or during life events such as reading, resting, relaxing. You're thinking about, for example, you've just had a big meal, you're sitting around a fire, and you're just sitting back and reading. Therefore, your eyes will adjust for closer vision, for example. And of course, your body is quite active, but it's more active in terms of digesting this big meal that you ate rather than activating yourself like your body in terms of exercise, like it would be on the sympathetic nervous system side of things. Now, a big part of what we're exploring here is sort of a complex story. We're exploring in some detail exactly how the central nervous system creates impulses that eventually reach targets. So remember that the central nervous system will be the brain and the spinal cord. So up above there, we're going to draw a portion of the brain, and specifically a portion of the brain stem, the midbrain. And we're going to skip over the pons and we're going to draw the medulla oblongata. Those are the two parts of the brainstem that are involved here on the parasympathetic side of things. We start with the parasympathetic side because the stories are actually a little more straightforward. Everything pretty much works the same way. On the sympathetic nervous system side of things, it's a little bit more convoluted than that. There's lots of exceptions here and there. So we're going to start off with a simple story first, even though the parasympathetic side is not as well known. So we're going to go ahead and label the spinal cord. Up at the top, you have the cervical branches, the cervical nerves of the spinal cord, and then thoracic, and then lumbar. But we're actually most interested in the sacral region, which is down at the bottom there. We're specifically interested in S2, S3, and S4 of the spinal cord. So the parasympathetic nervous system, up above, we're going to have a portion of the brain. And then down below, we're going to have the portion of the sacral spinal cord as well. So this is considered the craniosacral branch of the autonomic nervous system. On the other hand, when we deal with the sympathetic nervous system, again, we're going to go ahead and draw a brain, and we'll draw the whole spinal cord. But the only part of the spinal cord we're interested in are the parts in the middle. In fact, we're interested in the thoracic region and just a bit of that lumbar region. Basically, it's going to be T1 through T12 are going to be involved here, and then L1 and L2 down at the bottom. Thus, the sympathetic branch of the ANS is considered to be the thoracolumbar branch of the ANS. So again, the parasympathetic branch is considered the craniosacral, because those are the portions of the central nervous system that are involved. And the sympathetic side is called thoracolumbar because those are the parts of the central nervous system involved in instigating activity from these branches. So the central nervous system, then, we're replicating it on both sides. Of course, you know that you don't have two brains and two spinal cords, but it's actually easier in some ways to draw the targets in the center and then engage with how each part, the parasympathetic branch or the sympathetic branch, is engaging with each of them so that you can see, for example, you can readily see things like dual innervation, which we're going to be talking about in very shortly. So let's go ahead and list out our targets. Our target structures or organs or even organ groups are fairly broadly categorized here, so we can just kind of get a taste for how everything works. The eyes receive information both from the parasympathetic side and the sympathetic side. So do the salivary glands, so do the lungs, and so does the heart. These four structures have antagonistic connections from the parasympathetic side or the sympathetic side. So depending on which branch is more active in that moment, it depends on whether those structures are enhanced or excited or inhibited. Right in the center there, the sweat glands, the blood vessels, and the adrenal medulla are only going to be getting innervation from the sympathetic side. Abdominal organs is a big group, but the abdominal organs you can think of as being your digestive structures mostly. And then, of course, we have the bladder, the urinary system is represented there, and the genitals, the reproductive system is represented. It might be helpful. There's a lot of details that we're going to be covering, so it might be helpful, although you probably don't know all of the details about how all of these target structures work, you have some idea of how they work. So I will say that these are all dependent on either muscle contractions or on glandular secretion. So a good example of a glandular secretion would be the salivary glands. They secrete saliva. Another example would be the adrenal medulla. They secrete a substance called adrenaline. That's where the name adrenaline actually comes from. When you think about muscle contractions, you might be thinking of contractions of muscles in the eye, for example, the iris, or the muscles that control the fattening of the lens, the accommodation of the lens of the eye. Other muscles might be, for example, the blood vessels. There are muscles in the wall of the blood vessels that allow them to constrict, or when they're relaxed, of course, the blood vessels dilate, and that helps to control blood pressure and blood distribution around your body. So anyway, there's quite a lot of detail to it, and this is a big part of what we're going to be covering in the second semester of anatomy and physiology. So it's helpful to kind of have that big picture understanding that actually harkens back all the way back to chapter 1, or one of the very first lessons we learned in this class. So we talked about homeostatic imbalances, and we talked about regulating how things work, and we talked about the effector and the response of the body to things. And you may recall that we mentioned that everything that an effector does either falls under the category of a muscle contraction or a glandular secretion. And here we are. We're actually talking about those things. So although there's lots of details behind that, that's basically what it's all about. It's all about muscle contractions or glandular secretions. Each and every one of these targets is going to be doing one of those things in response to innervation from the ANS, either the sympathetic side or the parasympathetic side. We are going to be starting with our blue marker, and we're going to go ahead and draw some dots. These dots represent cell bodies and dendrites. And as you can see, we're starting with some dots in the central nervous system and specific regions of the brain. And we're also going to go ahead and draw those circles there. Those represent ganglia. In the peripheral nervous system, ganglia represent an area, a little structure, a little membrane-bound bulb, where a synapse can occur. So what we're going to do is we're going to draw a line, which would be an axon, between the cell body, which you notice is buried inside of the CNS, and then we're going to draw an axon terminal as a V inside of the ganglia. You'll also notice we're drawing a few lines that we're sort of leaving either free, or we're drawing little V's or axon terminals that are outside of ganglia. These actually are inside of ganglia, but they're not very well developed ganglia. They're going to be very close to the target structures. Okay, and then we're going to go down to S2, S3, S4, and we're going to do the same thing. 3 dots, 3 lines. One of them will be in close proximity to the abdominal organs, and then the bladder, and then the genitals. Let's go ahead and turn to the sympathetic side. We're going to go ahead and draw dots inside of each and every one of these nerves, inside of each of these boxes, so that it represents cell body and dendrites inside of each of those. And now we're going to draw an elongated ganglion. Remember, a ganglion is a place where synapses can occur. And it turns out that we have this structure called the sympathetic trunk. It's only on the sympathetic side. There's one on either side of the spinal cord. And the sympathetic trunk is basically an elongated ganglion. And so I'm going to go ahead and draw a sympathetic trunk on either side so that we know that it's there. So again, you're going to be looking for synapses to occur inside of either the ganglia drawn over here or inside of the sympathetic trunk. And we're only going to draw the one side, but it would be reflected on the other side as well. So let's go ahead and draw a line, and our first line will have a V at the end. And up above there, so we're going from T1 straight across into the sympathetic trunk. We're going to draw actually 5 up above and then 1 straight across. We're going to go from T2 and we're going to draw 1 straight across and another V there, another axon terminal. And then we're going to skip down some. We're going to go from T5 through T9 and we're going to draw some branches there. And now you'll notice that they come from different nerves, but they actually form a network together. And part of this network is going to dive down into this ganglion. But another part of it is going to actually go straight across and not even form a synapse. So this is a specialized connection, and we're going to come back to this lesson a little bit later. But for the most part, in both branches of the sympathetic nervous system, on the parasympathetic side or the sympathetic side, there are always 2 neurons, one leading to a ganglion and then one leading away from the ganglion. It's called a preganglionic neuron and a postganglionic neuron. We're going to draw some more down below here from L1, L2, and again, they're synapsing inside of that ganglion. But in the case of the adrenal medulla, there's actually a direct synaptic connection from the spinal cord directly to the adrenal medulla, which is very powerful. The adrenal medulla, I mentioned this before, the adrenal medulla is responsible for releasing adrenaline, epinephrine directly into the bloodstream. So to be able to have this direct synaptic connection into the adrenal medulla is quite powerful. It's a good way to release a big burst of adrenaline directly into the bloodstream and then have access to that basically circulates to the rest of the body. So I mentioned preganglionic neurons. Now let's go ahead and go over to the parasympathetic side and draw our postganglionic neurons. The first neuron in a row is called the preganglionic neuron, and then we have the synapse inside of a ganglion. And these are oftentimes on the parasympathetic side. They're very close to the targets. And then we have a postganglionic neuron as well. And you'll notice that they're all blue. And go ahead and take a look at the different targets that are happening there. You'll notice, for example, that the abdominal organs, it's a big category of structures, and they get input both from up above in the medulla and down below from the sacral region of the spinal cord. The reason we use the color blue is because these are cholinergic neurons. Cholinergic neurons are neurons that produce and release acetylcholine. And you'll notice that both the preganglionic neurons and the postganglionic neurons are both cholinergic. They both produce and release acetylcholine. Well, on the left-hand side, on the sympathetic side, there is one synaptic connection that is just like that. It turns out that when the target is the sweat glands, both the neurons are blue. The first neuron in line, the preganglionic neuron, which synapses inside of that sympathetic trunk, extends to another cholinergic neuron. So the preganglionic neuron is blue, and the postganglionic neuron is blue. They're both cholinergic neurons. And this happens to be an exception on the sympathetic side because all the other connections are actually going to be red. What red means is that these are neurons that actually produce noradrenaline primarily, noradrenaline or norepinephrine. Same thing. Noradrenaline and norepinephrine are the same thing. And they also may produce a small amount of epinephrine or adrenaline as well. Which, by the way, those are also the same thing. Adrenaline and epinephrine are the same thing. So what you'll see then is that the preganglionic neurons, for the most part, On the sympathetic side, the preganglionic neurons are cholinergic in blue, and the postganglionic neurons mostly are red, which is adrenergic, which means that they produce norepinephrine and epinephrine. Now you'll notice that the exceptional situations that we had, both the sweat glands and the adrenal medulla, only receive input from the sympathetic side, and the blood vessels are in there too. So all three of these structures, the sweat glands, the blood vessels, and the adrenal medulla, are only receiving input from the sympathetic side, which you'll see more clearly in a moment when I draw the arrow, when I draw the line directly to the blood vessels. But you can see right now that actually, if you look closely, and if you're kind of paying attention, that they receive no input from the parasympathetic side. So when we draw our lines, when we draw our synaptic inputs directly to the eyes and the salivary glands, what you'll see then is that there is input coming from the parasympathetic side and from the sympathetic side, dual innervation. So it's receiving input from both branches of the ANS, but they work at cross purposes with each other. For example, with the eyes, the parasympathetic nervous system would be more likely to cause the eyes to see close vision by accommodating the lens inside of the eyes, whereas the sympathetic nervous system, the sympathetic branch extending to the eyes, is more likely to allow the eyes to see distant vision by allowing the pupils to open up, allowing for pupillary dilation. So these kind of cross purposes there. The salivary glands are another good example. The salivary glands are more likely to be enhanced in producing saliva by the parasympathetic side of things because the parasympathetic nervous system is in charge of digestion, remember? And saliva secretion in the mouth allows you to digest things like carbohydrates well inside of the mouth. Whereas on the sympathetic side of things, in situations where you might be feeling excited about things or feeling some panic or some fear or something like that, your mouth might feel like it is drying out because the saliva tends to be thicker. It's more viscous when the sympathetic nervous system is involved, because what the sympathetic nervous system does to the salivary glands is it starts to reduce the amount of water coming in, so it's less watery and more viscous, the saliva. Probably better known would be the antagonistic innervation of the lungs and the heart. So the lungs are going to be enhanced in their activity. Your respiratory rate goes up when the sympathetic nervous system is in charge. Your heart rate goes up when the sympathetic nervous system is in charge, and the opposite when the parasympathetic nervous system is in charge. These two systems would be slowed down. There we have our red line going out to the blood vessels. Remember, the blood vessels are only receiving innervation from the sympathetic side. This allows for a very powerful situation called vasoconstriction, which controls blood flow and blood pressure in your body. Lastly, we have the abdominal organs, dual innervation, the bladder, dual innervation, and lastly, we have the genitals. The genitals are kind of an interesting story because yes, there is dual innervation, but in this case, the dual innervation is actually cooperative. So from the parasympathetic side, in order to achieve an erection, you would have to have a relaxation of the blood vessels that allows for plenty of blood to flow into the vascular spaces that allows, for example, the penis to become erect. But in order to have an ejaculation, you need stimulation from the sympathetic nervous system. So the dual innervation of the genitals is oftentimes considered one of the good examples of cooperative dual innervation rather than opposite moving or antagonistic or cross purposes. Now on the parasympathetic side, the preganglionic neurons tend to be quite long and extend branches almost all the way to the target organs. And then the postganglionic neurons tend to be quite short, sometimes right on top of the target structures. On the sympathetic side, it is the opposite. The preganglionic neurons are quite short, with the sympathetic trunk and those ganglia being very close to the spinal cord. And the postganglionic neurons are quite long, with the one exception of the neuron that goes directly from the spinal cord directly to the adrenal medulla. Remember, this is the one case where you have only one neuron and not two neurons in a row. In that case, that is actually considered a preganglionic neuron, although it innervates the target structure directly. It is quite long, of course, because it has to go all the way from the spinal cord to an organ. And I think the last fine point to put on this lesson before we move on to our next topic, basically, is that we are drawing only one neuron, but that single neuron always represents more than one, dozens or hundreds or even perhaps thousands. If you look, for example, at the one neuron that leaves the medulla oblongata and then eventually branches down to the abdominal organs, that actually represents the vagus nerve or cranial nerve 10. And that nerve is quite a large nerve. It has about 100,000 neurons inside of it. So although we're only drawing one neuron in all of these cases, keep in mind that it is just a representation of many more neurons inside of a much larger nerve. Now today we've only very quickly discussed the cholinergic and the adrenergic neurons, and you can see that there's quite a mix of them. Most of the preganglionic neurons are always going to be cholinergic or releasing acetylcholine. The postganglionic neurons are sometimes adrenergic or releasing norepinephrine and epinephrine. So I thought I would draw just a general, like 1, 2, 3 kind of a situation where you have a preganglionic neuron followed by a postganglionic neuron followed by a target tissue or a target cell. So let's go ahead and draw, 3 structures here. A preganglionic neuron, dendrite, cell body, axon, axon terminal, connecting with a postganglionic neuron, dendrite, cell body, axon, axon terminal, and a target cell of some kind. We're going to take a closer look at the synapse here. This is going to be a typical chemical synapse, just like we've seen previously in this class. We take a look at our presynaptic neuron is always going to have neurotransmitters that it releases. We're not actually drawing the vesicles here. We're not drawing a lot of details. It's just a really simple view of this. And that means that if neurotransmitters are released from the presynaptic neuron, then the postsynaptic neuron must have receptors for that particular neurotransmitter. And so we see that between the preganglionic neuron and the postganglionic neuron. And then again, from the postganglionic neuron directly to the target cell. The postganglionic neuron will also produce some kind of a neurotransmitter, either acetylcholine or noradrenaline. And therefore, the target tissue must also have receptors for that particular neurotransmitter. So in the next lesson, we're going to be dwelling and going back and kind of reviewing the information that we've learned today, but we're also going to be going further and saying exactly how is this information received. And it's going to feel like a lot of detail and almost like a lot of unnecessary detail, but in fact, this is how pharmacology chiefly works. We want to interrupt pathways, for example. We want to block receptors. Well, we have to know which receptors to block. If there's, for example, a painful stimulus happening, we want to be able to block that or maybe enhance it depending on the situation. Or if the heart is overstimulated, maybe we want to slow down the heart rate. So we have to sometimes look to neurotransmitter production, but oftentimes we're actually looking at the receptors on the other side of the synapse and saying, well, how are we going to block this effect from taking so much function away from the target tissue? So both the neurotransmitter is important and the receptor is also quite important.